Good morning, everyone, and welcome to another delightful event of Northern California chapter of HIMSS. My name is Nick Terrani, and I'm the chair of the Education Committee, and I have the honor of being your host for today's event. Before we begin, please allow me to take care of a couple of housekeeping items. For the next 40 to 45 minutes, everyone is muted. So if you have any questions, please feel free to go ahead and put it in the chat box or wait until the very end of the 45 minutes. We'll be more than happy to answer everyone's questions for the time we have available. Thank you for your attention. As you already know, the topic for today's event is precision medicine and the role of genetic mutations on comorbid conditions unveiled. Precision medicine is becoming a reality in medicine, recently driven by the need to understand more deeply the mechanism of cell injury and how that differs in individual based on their genomic makeup. This is evident in the gene and mutations responsible for adverse outcomes of COVID-19, as well as the many advances in the treatment of previously incurable diseases with cell gene therapies. Our genetic differences are one component of how we respond to diseases and other factors such as social determinants, diet, environmental effects, and the bacterial biome we all support. Today, we have three inspiring speakers for you. Dr. Cecil Lynch, Global Biomedical Informatics Lead at Accenture, as well as one of the valuable members of the Education Committee of our chapter. Dr. David Rowe, Global Chief Medical Officer and VP of Healthcare at Microsoft, and Dr. Clem McDonald, a Senior Investigator of National Library of Medicine. I would like to thank you again for accepting our invitation to speak on this panel, and welcome. Today's focus will be on the work being done in the science of genetics, as well as the technical support needed for modeling the messages to communicate the data and to get it in the hands of the physicians at the point of care where the data becomes actionable and meaningful. We will go through a few impactful use cases of genetically driven discovery and discuss what is being done to support the science with technology. With that said, if you allow me, Cecil, I would like to begin with you by asking you, in your opinion, what is your approach to identifying the comorbid genetic relationships?
Thank you very much, Nick. So as you know, there is a huge volume of data that is available from the NIH as well as other organizations around the world around genomics mutations. There are more than 700 million known mutations compared to the reference genome. There are thousands of biopathways that we understand, at least to some extent. And we understand the role of cross-gene mutations to an extent. There are many things that we do not know about genetics. So the way that we approach really getting to the heart of how the genes are related to a disease starts with understanding the sequence of that DNA molecule, parsing that into meaningful chunks that we can evaluate, and then doing the interpretation of those variants that we find relative to the data that we already have, and finding cases of repetitive mutations in individuals that do not have a known mechanism to then assign a probability that there is actually something related to the disease of interest around that particular mutation. So this process of discovery really takes an aggregation of data from multiple data sources, putting that together, linking the genes to the mutations, the genes to the biopathways to the proteins produced. And as you can imagine, this gets pretty complex. So trying to organize that information is part of the functions of the National Library of Medicine, the functions of the National Institutes of Health and others that make that data available to us. And then those like myself kind of pull that data, put it together, and then mine it. And that data mining is another process of trying to identify the specific mutations that are tied to that. Now, once you've gotten that, the process doesn't end there. As you talked about earlier, you have to be able to take that data, put it into the hands of genetic counselors who can link it together, discuss the findings with the patient and the family, so that there's a good understanding of what that mutation is and how it works. And then you need to reuse that data that you've newly identified to get into the hands of the clinicians at the point of care, so that the next time a patient with a phenotype that looks like they may have that, they're thinking about that new mutation that's discovered, or the science around existing mutations that perhaps isn't at the top of their medical knowledge, because it's a 1 in 50,000 disease that they may have passed over in medical school, with a 3-second blurb, or never even heard about it because it's so rare. But those rarities are the things where our genes and the interpretation of those genes is absolutely necessary to get therapies for these rarer diseases and these data sets that have numbers of the hundreds or small thousands of patients that we previously could not offer any type of therapy. So hopefully that answered your question about the approach.
Fantastic, thank you so much. Where do you think the future is as far as precision medicine in maybe 5 years?
Well, it's hard to say because we're changing so quickly. So I think that, if you follow this area of science, there are, even in the approaches that we've developed in the past, so CAR T therapy is a good one. It's a few years old now, but it's finding new therapy intervention protocols that can be used in CAR T therapy. We have cell gene therapy now that's really modifying at the cellular and genetic level, replacing a particular mutation in a diseased gene, and then having the body take that in and replicate that in the bone marrow and treat the disease internally from that new mutation that you have corrected in the genes of a particular cell type that can then replicate. But recently, and I'm talking about in the last 60 days, there were completely novel ways to use these. So there's a type of progressive blindness that was incurable in the past that disease is now treated by directly injecting these mutated from our perspective, from the original perspective, mutated in a way that we've corrected that defect, it injected into the eye, and that has been successful in treating a previously completely untreatable type of blindness by having the cells in the eye directly reprogram the synthesis of new retinal cells by that new mutation correction in the cell line. So these are some of the things that are there now. I had a conversation with one of my colleagues at Emory last night. We're running a project there on studying Alzheimer's disease, and he had just been back from an NIH study section and had heard things that are completely new to both he and I, that are not part of this topic, because it's a new approach to modifying things at the chromosomal level that then cause allele representations of that to go from the embryo on forward. So I don't know where we're going to be in 5 years. It's impossible to say, but I can tell you that the progress just in the last 2 years has been phenomenal in this area. And I fully believe that we will be able to treat diseases like diabetes, not with insulin or oral pills to change the way we metabolize in our liver, but we will not get it because we won't have the oxidative death of cells in the pancreas that produce the insulin. And we will have corrections for those types of things. So diseases that are prevalent and affect, Clem's probably more accurate on this than I am, but the idea is that in the United States, the population of diabetics and pre-diabetics is about 110 million, or a third of our population is affected by that. So when you look at the changes coming in precision medicine to be able to completely wipe out diseases of that magnitude, it's amazing. If you look at cancers and what we've been able to do there, when I started practicing, we had about 25,000 cases of cervical cancer a year, right, in women. We now have less than 2,000 a year. And why? Because we've been able to model a vaccine that actually inhibits the replication of the major virus that causes that. We have trials running at the NCI right now on treating the most common type of breast cancer that is completely able to prevent that in a mouse model, that all of these mice get this particular type of lobular carcinoma, and we can completely prevent that and actually reverse it, even if they have it in about 40% of the patients. So that's what to expect as we go forward in the next few years, is treatment of diseases that there is no treatment for today, and new treatments where our treatments are ineffective.
Fantastic, very insightful. Thank you so much, Cecil. If you allow me, David, if I may ask you, where do you see, or how do you see the role of technology vendors in precision medicine?
One of the challenges that we have with precision medicine is that there is just so much data out there, and it all needs to be interconnected. As Cecil had described, what we found is that, first and foremost, a human can't do that. It's just too much. We found that spreadsheets are unable to provide that level of connectivity. So we're really talking about platforms, cloud platforms that have that compute capability, ability for us to be able to build AI algorithms and allow us to be able to do this in a rapid amount, or a very fast amount of time. That's something that is just the underlying platform. But where we're actually now starting to recognize the need is that there is a need for collaboration and data sharing that needs to occur within organizations. Oftentimes, as we talked about, these are one organization may be lucky enough to be able to capture a certain number of patients that will have a certain condition, but oftentimes it requires multiple organizations, not just even within the same country, but also within the entire world collaborating. And so what we've been now looking at are these new models in which you have organizations sharing data in different manners, and creating larger data sets and platforms that allow us to be able to draw insights from this. And so that requires a partnership between technology providers, researchers, pharma life science organizations, to be able to create different types of mechanisms for allowing this data to be shared. I'd say the traditional method has been around creating a single, very large data set that's, allows researchers to all work within this environment. A great example would be some of the work that we've done, more recently with the Truveta Coalition, a large number, 17 large health organizations all working together to share data into a common platform. Another great example would be Terra, another Microsoft partnership where, looking at how all this information can be brought and centralized. But we're also starting to see federated models where data can then be brought into different areas where people actually don't need to invest or share all their data into this common platform, and allow for algorithms to be run on top of that. So technology enables that. It's so, as we think about the possibility of how technology can be useful, it's storage, compute, artificial intelligence, building tools on top of it, but also creating models that allow for effective data sharing, privacy, and security.
Fantastic, thank you. In your opinion, who are the major consumers of the platform technologies in genomics today?
Well, I'd say largely because much of this is still an evolving area, we see a tremendous amount of interest in research, R&D. And R&D falls into the life sciences companies that are building the products, the technologies, and the different innovative ways for us to learn more about it. But also it's the academic medical centers and the research organizations across the country and the globe that are very interested in this. And then again, as I mentioned before, and also looking to ways that they can collaborate. And so you'll oftentimes see government agencies that will be very interested in terms of being an aggregator of a lot of this information. So the NIH would be a great example, but certainly within the UK and other parts of the world, we've seen tremendous amount of interest from government agencies to help build and support this. So it's really all those, it's public, private, but in large part, it's because of the fact that we continue to evolve our technologies. We learn from it and we build off of it.
Fantastic. Thank you, David. Clem, in your opinion, what role do you see standard bodies playing in interoperability of genetic data?
Genetic data? Well, I have to apologize first. I have no polka dots. I'm the only one with no polka dots. My closet didn't have them this morning, so I apologize.
You look great. You look great.
Well, it's complicated. What I'd like to highlight is the difficulty, the barriers. So the conflicts between privacy and sharing are a challenge. So at NIH, I think they're coming up with a solution. I'm from NIH, using a mechanism whereby users can access a system, and it's well known who you are. So they pass around encrypted sort of passports that let it get from one system to the other, but people are completely verified as to their identity and what they're allowed to do. And there's a lot of restrictions. So there's sort of a tension here. And I was in another meeting earlier today where there was a question about being able to share data with intensive care unit data. And one of the hospital participating, smells like they're going to back out. They just don't want to run any risks. So we have terrified administrators and we have eager researchers, and we got to find a way to blend the goals.
Fantastic. Now talking about NIH, would you share with us what NIH is doing with regards to standardize the data across the multiple aggregates?
Well, NIH has a huge system actually, which we're working with. There's a billion records from about 17,000, maybe now 18,000 studies, where this is the phenotypic data, like the lab tests and the findings and the survey instruments. And then they have behind that, they have in some cases complete genotypes. In some cases it's more targeted, but it's genetic data. But it's a challenge for a lot of different reasons. First, it's huge, it's slow. We're actually implementing it we're trying to implement it in FHIR as we speak. Not the genetic data yet, we're not prepared for that, but the phenotypic data, and provide a mechanism where researchers can search across various studies with the same variables, and also to standardize those variables on the way. So currently, researchers can get into it with this very strong protection, and they can download the data, including the genetics, but very restricted. We'd like to be able to let them go across studies so that you could broaden the views, get different diseases, compare them, et cetera. I think it's going to work. They have a very sophisticated mechanism for controlling access. There were still challenges with it. We're working through some of the issues and problems.
Thank you. Fantastic. Cecil, if I may ask you, how are your clients using the genomics related comorbidity data? And perhaps you can elaborate by some use cases.
Sure, absolutely. So maybe it would be kind of helpful to show what we mean by these shared mutations. I have some people are not as good at auditorily picking this up, especially if that's an area. So let me just kind of quickly show a slide that will give you an idea about what I'm talking about here. So
okay. Thank you.
If I may. So when we, let me just go into slide mode here. If I can get my pieces of it. So when we talk about shared mutations, this is really a view around a group of diseases that have lots of shared mutations, the connective tissue diseases. So the purple basically says there's a mutation. This is kind of how we talk about mutations, this RS number. And those specific mutations you can see are shared across, these three from rheumatoid arthritis, are shared with the Sjogren's syndrome, are shared with lupus, or shared with systemic sclerosis. So it's no surprise that, if you actually mine large data sets, you'll see that a person who has psoriatic arthritis or who has lupus is 4 to 8 times as likely as the general population to have yet another one of these diseases. And that's because of all of these shared genetic mutations that an individual is likely to carry from one or more parents around these disease states, and they tend to accrue over time. And that's what we talk about from a perspective of shared mutations. But there are other diseases that you don't really think about. And if you have enough data to look at this, and we have about 55 million patients with complete medical records on our platform, then you can start looking at things that are really bizarre, right? So we see, I've highlighted here, St. Louis encephalitis virus infection. And what you're actually seeing on the screen is when I select this particular comorbidity, I can see that it occurs about 420 times as likely in autoimmune diseases as it does in the general population, right? And what this is showing is that all of these gene variants that occur in these connective tissue diseases also allow infection of this virus in a human. If I reverse this around and I looked at St. Louis encephalitis virus, there are about 358 of those in our platform. About 85% of those patients have lupus as well. So that's just an example of how mutations that we acquire genetically are shared across different comorbidities, and some that you wouldn't even think about. This particular one is not in the literature anywhere. It's kind of something I've discovered recently just by mining our data. But we'll get that published. And this is the example that I talked about earlier about where you may not actually know all of this because as David pointed it out, it's so vast, the amount of data, that no human can pick it out unless you have the tools that allow a quick and easy visualization of this. And those tools have to aggregate these hundreds of millions, billions of data points. It's not something you're going to do on your laptop. So let me kind of stop sharing there. Hopefully that gave you a visual representation of kind of what we mean by shared comorbidities and the drivers of those from the mutation perspective.
Very good. Thank you very much. Fantastic. We do have a good question from a colleague of mine, actually. And this question is addressed to any of you as you wish to answer. This is regarding the accessibility to precision medicine. Will this breakthrough approach be affordable either through insurance or government programs to all? Please comment, any of you.
I can take a shot at that. I think probably Clem and David will have their own comments.
Please,
there,
please.
But certainly, I work with a number of these genetic processors, and the prices have been dropping significantly. And I think if you back up to the late 80s, early 90s, when we were sequencing the whole genome, Clem can probably talk about those numbers better than I can, but it was in the hundreds of millions to billions of dollars to sequence that first whole DNA sequence of an individual. Our ability now to take a serum sample or a tissue sample and run the sequencing on that is roughly in the range of $1,000 to $1,200, right? And that price is dropping. But just looking at where we've gone in the last 20, 30 years of being able to more cost effectively understand these DNA mutations is pretty amazing. Now, sequencing is not the only cost. You've got to actually store that data. You've got to process it, but at an individual level, it's there. And to the point of kind of, where we're at on the cost and access to that, if you look at specifically what is being required right now to pay for some of these extremely costly medications, one of the requirements oftentimes is that you've got to prove that the patient has that condition before you are going to give them one of these medications that can run in the $100,000 a year price ranges, right? And so a couple of good examples of that is a treatment for colon cancer, advanced lung cell cancer, melanoma. It's called Keytruda is one of the brand names, but there are a couple of these in that line. And those drugs are extremely expensive. And so before you can give one of those drugs to a patient with one of these cancers, you need to do genetic testing on them to see that they carry enough of the mutations that drug is likely to be effective in that disease. Because otherwise you're paying for a drug that may not be effective, and insurance shouldn't be covering those things. And patients shouldn't be subjected to the side effects and the hope that is engendered by getting these drugs when it's not likely going to be effective. Another good example is in a hereditary disease, hereditary angioedema. It's a difficult to diagnose disease. It is one of those 1 in 50,000 kinds of diseases, but once you have found a patient that looks like they have this disease, the next step before anybody is going to pay the $70,000 or $80,000 a year to treat that disease is to do the genetic testing and prove that they have it. So these tests are available. They're available more selectively where you need them to prove it. Another one is called ATTR, a type of cardiovascular disease that is kind of, you hear about this one, people know about it, they don't know what the disease is, but you read about the 23-year-old athlete on the college football field dropping dead on the field, a completely healthy kid. Well, the vast majority of those cases are because of this particular cardiac condition. That's a genetic condition. And there is a therapy for that now, but you need to know about it early enough to make the therapy useful, which means you need to have effective ways to find the phenotype, which is genetically driven, being able to understand the comorbidities that appear before you get that disease. And then you need to do the genetic testing to confirm that disease. And so it is real today, and it's what's there. Another quick example is if you look at where the FDA is right now in the labeling drugs. So there are 350 diseases right now, 350 drugs that have some form of black box warning or recommendation for screening or special population requirement. Many of those are genetically driven. And so the FDA is now getting there where they're making sure that the label carries enough information to you so that you can understand and have that conversation with your physician to see if you're carrying a defect in a particular enzyme needed to metabolize that drug. And that's an area that we call pharmacogenomics, is understanding how we metabolize these drugs in the body. But there's a whole nother area, right, that is coming up around understanding the biome, so the bacteria we carry on our body, and how that might affect metabolism of specific diseases. So that gives you an example of kind of, one, the cost is getting down to where we're ordering these more routinely before we prescribe drugs, because the drugs are more expensive than the genetic testing, right? And you want to make sure they're effective. And we're going to see more and more of that as time goes on.
Yes, Clint, go ahead. Perhaps.
Well, I think that, we're talking about human genetics, but I don't think we should forget about infectious disease genetics, which has had, it's in the paper every day. It's a spectacular effect. And, this whole idea of sequencing a virus and then sticking it into the body as a messenger RNA, hoping that it'll get caught up by the cells and then make that protein and then protecting a patient. It's almost, it's wonderful, and it's a surprise in a lot of ways. They had to do a lot of things. They had to cover the messenger is really very fragile, and it gets eaten up immediately by the blood. But they put some fat around it, they tweak it, they did cool things that made it get into the cells. And and here we are with 80% of the people in the US having at least some shot vaccine, almost all of them based on the same technology. And then the diagnostic opportunities with genetics in the virus and bacteria, they can find little teeny pieces of the bacteria which they couldn't find before, proving someone has a disease or not. You can do things where you get someone coughs up some sputum and you can test for 20 or so viruses or bacteria at once and know whether they're sensitive or resistant. So the the big splash so far has been in infectious disease, and what we're anticipating, and it's not so expensive. I mean, you can do some of these tests for $10, $15, maybe 20. But when for human genetics, the promise is huge, and we're still working through it. We got a lot more DNA. I mean, a lot more nucleotides than the viruses do.
And I think what you're hearing from both Clem and Cecil is that precision medicine is a very heterogeneous topic. And when you think about what we do today in healthcare and what we do in medicine, it is very imprecise. We take a patient that comes in, and based on some general factors, this is a person with hypertension. They're of a certain ethnicity. I'm going to prescribe them as a first-line drug, this medication. What we do is we base it on statistics, but the reality is we would be far better off if we had much more specificity in the drugs and the medications and the treatments that we offer to individuals. And when you look from a cost perspective, let's not just look at the cost of the medication, but let's look at the cost of the fact that you have, from a population perspective, a large number of individuals that are being inappropriately being treated versus those that are potentially not being treated as effectively as could be. If we were to improve the efficiency of our healthcare delivery so that the right people got the right medicines and people didn't get the wrong medicines, we could improve the overall cost for the systems in general. So I think we have to start looking at the total cost of care for populations as we start deploying these treatments. Now, the reality is some of the treatments, because they're newer, they're more, and if they're not subsidized, they will be more expensive. But in the long run, if you look at across over time, it'll diminish, and as we learn for ways to make it more efficient, it'll diminish. But also looking selectively at the total cost of care across a person's lifetime. Some of these may be life-saving, some of these may be essential. And so we have to weigh all those factors when we look at whether or not some of these drugs and treatments should be promoted, should be subsidized, should be paid for by the payers and other organizations. And I think that oftentimes we focus too much on just the price tag of that one particular item. It's really the total cost of care.
Fantastic. Following up on the same question, my colleague Jeff is asking if we are going to start having some type of a preventive genetic screening to identify potential or impending disease states, in your opinion.
Let me say that I think that, on the topic of precision medicine, there are lots of ways to approach this before you get to the gene, right? So, I'm often asked about what does the future doctor's office visit look like? So I think, one of the technologies that a lot of us that fly a lot, you walk in and you go through the clear line and you're identified by your retinal scan, right? Because it's unique to you. There are many diseases that show up in changes in our retinal configuration. So I think that, when you start looking at how medicine is going to change from a precision perspective, you're going to walk into your doctors. There's going to be a cheap way to do a retinal scan. You're going to learn a lot about whether they have hypertension, what does diabetes look like, they have other vascular diseases. You might have them stick their finger in and do a quick kind of Apple kit scan of what your cardiac rhythm looks like. It can look for things for diabetes. It can do types of that. And then you may go from that next step saying, well, you've got a few of these things that show up on our screening precision tests. You need genetic testing, and it'll be available pretty rapidly. I think you'll start seeing it first show up in your local Quest or LabCorp office, that you'll send them over and they'll get a screening test, and it'll be back in 45 minutes or so. And then eventually they'll get down to a point where you're doing it in the doctor's office. And that may be 10 years from now, but we're going to see this whole field progress, not just in the genetics of precision medicine, but the screening tools to decide whether you need processing of that genetic medicine.
One other thing that, when we think about screening, and we oftentimes think of genetics as being sort of the cornerstone, but genetics is like a blueprint. It allows us to understand whether or not you are more susceptible to receiving or being able to get a certain type of infection or disease. It doesn't necessarily mean you're going to have it. Now, there's oftentimes a high correlation to that, but there are other factors. But as you think through this process, a lot of what we're interested in as well is whether or not you are early in the stage of getting it. So maybe you've got the blueprint, but you've also now started secreting or excreting proteins or other types of components that are from the cancers or the other areas, where now we can, through the precision medicine, we can pick these things up when it would have been extremely difficult before. And that's another aspect of the screening. So it's not just about the blueprint, but it's also about, can we identify early detectors or early markers of disease itself before we can get to the point where the disease manifests and extends and spreads. And so a large part of what we're trying to do is we think about it on the spectrum of all the tools that we have available. We want to start from the very beginning. You have the blueprint, but then we want to get to the point where some people, we might be able to look and identify certain aspects throughout their disease course. And then as they go into the course of, let's say, have a known diagnosis, figuring out ways that we can make the treatments more specific and targeted. There's just so many aspects for how precision medicine can be applied in healthcare. It's not just a one size or one model. It's many different models.
No, I agree. And I'd like to bring up just a more maybe exotic or futuristic part of it. If we're talking about cost of medicines and cost of care, sickle cell disease, we've known the genetic problem, one single amino acid, for at least 40 years, maybe 60. And we're just now maybe getting to the point where we can fix it. And that would be miraculous. I mean, some of the fixes are actually by faking it to make more of a different kind of hemoglobin. But then you got whatever it costs to fix it. You've saved the whole life of medical expenses and pain, et cetera. So there's huge leverage. And I think that's going to continue. And when we think about this messenger RNA for vaccines, it's also got other potential. You could probably replace some missing protein or missing factor. And it's now being studied for peanut allergies. So what I don't know how it's turned out, but what they do is they give them messenger for a peanut protein, and then they lose their allergy because it overwhelms their allergy system. And they're still studying it, but I think we're going to have just wild new possibilities, and then it'll become important to find that mutation that you can fix either by inserting through a virus the correction or repairing a messenger. Who knows? It just opened up so quickly with this with the vaccine studies that I think we're going to have a lot of fun watching the stuff unfold.
I think so. It's a very powerful conversation and complicated, very powerful. Thank you so much. This has been very empowering. I would like to go ahead and invite the audience to go ahead and post your questions. We do have one question coming up. Let's see. Next question is, given the capital requirements for R&D in this field, what kind of opportunities exist for technology startups with smaller funding and or with lesser reach?
Well, let me jump into a little bit. I'm not part of this. I don't do that sort of stuff. But there's examples, including the guys from Germany who did the Moderna. They just had a belief and they dug in and did it. They had a struggle getting any kind of capital funding for it. So I think good ideas, you need good ideas and you need fanatical followers of their ideas. And I don't think you have to necessarily be big capital expense to start with. Anyway, good ideas and intense belief in the ideas.
I think the industry is moving closer towards, now we're starting to recognize the real value that precision medicine can play. I mean, the Clem's example of the mRNA vaccines is a great one because people can relate to it now. They see how this can be done in a way that can be launched within a year's period. I mean, it's just amazing that you think of being able to come up through that process, get through the regulatory, and now have a product that's on the market. People now recognize that this is something that is not just going to be sitting in R&D labs for years, but it's something that can be brought to market. So I would say that the environment's a bit different now than it was perhaps a year or 2 years ago. But at the same time, there are mechanisms where a lot of times the infrastructure that's required to manage this is something that is extremely difficult for startups. And at Microsoft and Accenture and other places, we try to provide a lot of that infrastructure so that startups are able to focus on some of the things that they're really good at, which is the science and understanding and testing it. But when you're talking about how do you secure the data, how do you enable data sharing, how do you make sure that this information is being analyzed with the compute power, that's infrastructure that doesn't necessarily have to be something startups have to invest in. So there's now new ways that startups can leverage the power of the larger organizations, but at the same time, they can be nimble enough to be able to build off of it.
Very good. Cecil, any comment?
Yeah. Unlike Clem, this is my day job. I actually work with a number of different venture funds external to us, and we have a venture fund that we invest in these small startups as well. But I do this for a number of pharma companies that have venture funds. And my job is to actually go out and do the due diligence for these. How real is the idea? Is it scalable? Does the technology infrastructure allow interoperability across other systems they're going to need to interact with? That kind of thing. And I would say that I see a lot of these small startup companies, not just in genetics, but definitely in the genetic side. But when you think about precision medicine, there are many other areas, right? So there's AI around better evaluation of cellular structures for cell gene therapy, or for cancer detection, or for things that get down to that level of precision. And there is a lot of activity in that area. And especially you will see the big pharma clients of the world who, of course, they have their regular business, but they're also big investors in technology startups, looking at those types of technologies as ways to not only help healthcare in general across the country, but also to provide inputs that are going to help them build better drugs over time. We also see Silicon Valley VCs that are asking us to take a look at these firms, and there's a lot of money out there looking to invest in these areas. And I think, Clem, I just want to say that you brought up an excellent point. So I had one of my colleagues in Accenture when we were talking about looking at specifically at investment in a small genomics startup company, and they were saying, well, genetics is still very small, and the effect of that's very small. And I put into the chat window, I said, you're probably walking around feeling safer because of a vaccine you got that is all about genetics, right? So I'm glad you brought that up, the fact that this isn't just about human genetics, but it's about our understanding of genetics in general for building these kinds of things that are all about precision medicine and how we work with those.
I have one question from the audience, and the question is the role of EMR contribution to precision medicine.
Well, a lot of the information is captured in the EMR, and so it's important that we think about how that information can be shared in a way that's interoperable, allow us to be able to provide the tools and the open APIs that allow for that exchange. I think we're getting to the point where that is becoming much more standardized and people feel comfortable leveraging FHIR, for instance. We are recognizing that there's a lot more that could be done in terms of how EMRs can be much more interoperable. But I feel like we're heading on a journey, everyone, EMRs included, around the fact that we are looking to share data in ways that allow us to be able to draw greater insights. A lot of what we're talking about here can only be done through data sharing. It's difficult, if not impossible, for one organization to have enough information and data points to be able to draw insights on the fact that you've got like St. Louis encephalitis. I mean, how many patients have St. Louis encephalitis in a database unless you're doing data sharing? So EMR will be a central piece to this, as will disease registries and other types of opportunities for us to be able to enable that data, that amount of data sharing.
Fantastic. Thank you. So question for both Dave and Cecil, with regards to the IP for startups, how do they feel protected when they're working with companies like Accenture and Microsoft?
I'll start with that. So the first thing that, that I do when I have somebody hooks up with me on LinkedIn and said, do you mind looking at this idea? And I said, sure, I'll be happy to, but don't share it yet. Here's an NDA, right? So that they're comfortable that there's a non-disclosure agreement. And in our NDAs, standard ones are saying, our discussions with you are private. There's no IP. We are going to guarantee you that we're not going to use your IP for anything. Any discussions with you are protected, right? So starting out with a simple thing like making sure you're not having that discussion with this small startup until they've executed an NDA that basically protects them, right? And then from there on, I think I could probably speak for Microsoft as well. I work a lot with David and our technologies are on their platform. You don't see reputable companies like an Accenture, a Microsoft taking IP from anyone, right? They're really about integrity as much as they are about innovation. And we all work with tons of small technology vendors. Microsoft is massive. We have 650,000 employees with more than 300,000 developers. We could probably build anything we want to build. We're not a product company. We're an integration company. And when products come to us, we talk to them about their product and how to get it into the market. So that fear level is a little different with us. We're not going to replicate their products. We don't do products.
Well said. Dave, any comment?
So when organizations, whether they be small, medium, or large, partner with Microsoft, we are very transparent that we do not compete. We are very transparent about our policies around data management. We do not monetize data. We are here to protect the data and to empower our partners, whether they be small, medium, or large, to be able to achieve their goals. A large part of that process is building tools and that lower level of infrastructure that's required to be able to be much more efficient and effective. And then they can leverage those tools to be able to do much more. And so to one of the points that Cecil was mentioning about what kind of things can these technology providers like Accenture and Microsoft offer, an area that we oftentimes have found to be of incredible value is use of artificial intelligence tools. Microsoft's invested a tremendous amount into natural language processing, whether it be voice, text, image analysis, you name it. All of those are tools that we oftentimes are able to readily make accessible to our partners so that they can leverage them. A great example would be the text analytics for healthcare. Now, what we're doing is we're looking at information that's in an unstructured format. It could be in a database, but more often than not, it's often in progress notes, or it's in reports, or it's actually in the published medical literature. How do we synthesize all that information and combine that with the patient-specific information to be able to draw meaningful insights? That's a very difficult challenge for startups or even any organization to do. But now you've got tools that you can leverage on top of that. So we're really excited about partnering. We even have a Microsoft Startups program that you can enroll in. I can put the information in the chat window. It allows people to get that jump start in terms of working with us.
Fantastic.
I would point one other thing out very quickly, is the fact that you can go to Microsoft's GitHub and download a ton of accelerators and useful tools that are licensed under very open licensing that you as a startup can start with that. Same thing with Accenture's GitHub. You can go to Microsoft GitHub, Accenture's GitHub, and you can get very advanced analytic tools under Apache open licenses. And Clem hasn't said anything yet about all the work that he's doing on survey instruments and things that are available at the NIH that they're investing in to kickstart the capabilities of startups and everyone else, including us, that use the tools that Clem produces.
Well, there's one other source of data that isn't literally medical record I'd like to kind of highlight, and that's Medicare. Medicare has a system now where one can apply to use a selected subset of Medicare data for a particular purpose, and then but the data all stays on the Medicare machine. It uses their software, and so they're a little more comfortable with that than they were historically. I tried to get data when I was in Indiana about 15 years ago, and it took like 5 years, and I never got it. You know, they wanted the firstborn, thirdborn, and sixthborn child. I didn't have enough kids to kind of get any data. So, but it's pretty open, and it's really powerful. And we've done a lot of Cox regression analyses on numbers like 3 million. And we can distinguish between the effects of drugs which are not like ACE and ARBs are both thought to be good for stuff, various kinds of things. But the ARBs are really topping out the ACEs once we got to 3 million patients. So there's interesting things. That's medical record data in large. It's not as rich as medical record data yet. I hope Medicare will start pulling in labs and everything like other insurance companies do. But anyway, that's what it is now.
Very good. Clem, talking about Medicare, are you aware of Medicare working on any type of CPT code program for the prevention screening?
Who you addressing?
Clem.
I didn't hear the words. CPT?
CPT code. Are there any CPT code coming for genetic screening?
Okay. Well, CPT codes are everywhere. I don't know that we have them in the Medicare database, though.
Okay.
That's an American Medical Association.
Yeah, we've got ICD, and we've got drug names, common generic drug names. CPTs are what the all professional bills go under. And I think they are in there. Yeah, I think they are in it. Never mind.
Very good. Excellent. We are getting very close to the end of our program. I sincerely would like to thank Cecil, Dave, and Clem for joining us and taking the time to speak for us at this event. Thanks again, and I'm sure we all enjoyed it very much, and the recording of this event will be available on YouTube. With that said, I would like to invite everyone to participate in our future upcoming events. We have wonderful events coming up. Please go ahead, feel free to visit our website, Northern California HIMSS.com, and we do encourage you to get involved and participate. We have various committees that actually could easily enjoy your help, including my own committee, education committee. So please feel free to join us and participate. With that said, I would like to thank everyone for joining us and have a fantastic day. Thank you.
Thank you.
Thanks, everyone.
Thank you.